backward, forward. Let's try this.
Hello, my name is Gert Storm. I'm chairing the next session on the usefulness of nanomedicine for infection and inflammation treatment and imaging. And I would like to introduce you to Gerrit Borchard from Geneva. I know him very well, and it's because his first name is the same, Gerrit. And Gerrit means, maybe you don't know that, Girard in French. It means a strong spear fighter. So I think we will have a very active lecture and an active lecturer. And it starts with the title, Less is More. And there's more to that. And I would say more or less, that can be true in nanomedicine.
Okay.
Please.
Dank Geert. Ladies and gentlemen, it's a pleasure to be here. It's the second time at CLINAM. And to stay in the picture that Beat created, I brought a couple of new family members to the floor for this meeting. Very happy to be here. So this is a small study that we did actually on the development of DNA vaccine delivery systems, if you want. Most of the work was basically done by Johanna Pechheim in collaboration with a couple of friends. And she also has a poster here, the poster number 35. If you're more interested in this study, you can get more information on poster 35. Now, to make a long story short, this is, by the way, the look from my office in Geneva. To make a long story short, we were working on DNA vaccines against tuberculosis for quite some time. Here you see the pDNA that we were using. It's expressing the AD85A antigen, which is supposed to give you a sort of immune response against Mycobacterium tuberculosis. So what we do is we transfer not the protein, not the antigen, but we try to transfer the genetic information of this vaccine, of this antigen, to the host or to the vaccinated person. Now, we've been working for a long time with nanoparticles, and especially chitosan and chitosan derivative nanoparticles for delivery of DNA vaccines. Formerly doing stuff where we had aggregations between, or condensations between the DNA, the negatively charged DNA, and the positively charged chitosan or chitosan derivatives, forming nice nanoparticles. However, the problem there was that these aggregates did not dissociate well, such that we had some problems with the transfection there and the expression of the antigen in the end. So what we thought would be a good idea to preform these particles with using this chitosan derivative, trimethylchitosan, and do a condensation together with chondroitin sulfate. It's a negatively charged polymer, which you find in the extracellular membrane, forming these nanoparticles that you see here for the TMC nanoparticles. So these are TMC chondroitin sulfate nanoparticles of a certain size, usually about 300 nanometers, polydispersity, and a positive zeta potential. Now, when we adsorb the DNA to the surface, in order to have this presented here at 10, 50, and 100 microgram, we ended up with these sizes, with these polydispersities and zeta potentials growing more and more negatively when we increase the charge here. Now, here you see, as a TEM picture, a scanning electron microscope picture, you see these nude particles, and if you apply the 50 microgram, in this case, I think, to the surface of these nanoparticles, you see what's happening. It's changing the morphology, the size, the shape changes quite significantly. What you don't see here is that how much of the DNA is actually adsorbed to the surface of these particles, and how much of the DNA is actually free in the solution. Now, what we then did is that, what you have to know is that this plasmid DNA, of course, contains CpG motifs, which is a TLR9 ligand. So these, basically these plasmid DNA are ligands for the toll-like receptor 9. Plus, we incorporated in something like a dual approach, muramyl dipeptide, which is a ligand for an intracellular pathogen pattern recognition receptor, the NOT2 here, which included into the nanoparticles to have a dual approach of two pathogen pattern recognition receptor ligands in one go. We did all the physicochemical characterization, transfection studies, and so forth, but I'd like to focus only on the in vivo studies, and here only on one study. That's the experiment 2. You see here that we had 3 different studies in order to look at the efficacy of these DNA vaccine systems. In this second study here, you see that we had like 50 mice, 7 groups, 8 mice per group in order to reach a certain significance here. And what we did, we looked at the serum IgG and the T cells on restimulation interferon gamma. This was the in vivo schedule. You see that first injection, second injection, third injection, like every day, after 3 weeks, after 42 days, we sacrificed the animals on day 49. We did bleeding for this second experiment, serum, to look at serum IgG in terms of ELISA, and we did the T cell analysis and interferon gamma ELISpot. Now, this experiment 2, what were the aims? Now, the aims were that in the first study, we used a concentration of DNA of 100 microgram, I think, per dose, or 50 microgram per dose, sorry. And in this, we wanted to reduce the pDNA in order to see what this effect might bring. And then the second aim was to determine whether the addition of this NLR2 ligand activating the inflammasome would enhance even a Th1 response. The hypothesis was that lower amounts of pDNA would absorb more efficiently to our nanoparticles, so more of these DNA vaccines would be adsorbed on the surface and less free or slightly bound to the particles. So there would be difference between pDNA alone, which was a control, and TMC DNA would be more obvious. And the other hypothesis was that the MDP, this NOT2 receptor ligand, would further enhance Th1 responses for TMCA. These were the groups that we used, pDNA in PBS, TMC DNA, TMC DNA particles loaded with MDP, and we had a positive control here, the BCG vaccine, and a negative control, which was PBS. When we looked then, after the in vivo studies, looked at the ELISAs, looked at total IgG, we were disappointed. Because what you see here is that basically P and DNA gave us higher total serum IgGs than all the other groups. Just naked pDNA. So what we said is now, is that some suppressing effect of the adjuvants? So what did we do? So we had to look and do some troubleshooting, do some quality control of the DNA vaccine that we were using. First of all, we checked whether the genetic information for the AG85A was still in there, and we did this, and we had this report. Everything was peachy and fine. And then we looked at also the ELISAs, and there repeatedly we could see, okay, the protein is expressed, so we do not have a problem with the pDNA, so everything is fine in this regard. Now, then what we did is we looked at interferon gamma ELISpots. So we took the splenocytes from the vaccinated mice in this case, we re-incubated them with the protein AG85A, which we obtained from source, and then we looked at interferon gamma in terms of stimulation of these splenocytes, and we saw here that the T cell analysis did show an adjuvant effect for the TMC nanoparticles, and this was even enhanced by the addition of MDP, this NOT2 receptor, and that was quite significant, as you see here. So then we checked the, not the total IgG, but we checked the serum IgG subclasses, the IgG1, the IgG2b, and IgG2c, and there we saw a quite different picture. The pDNA alone gave us high titers of IgG total. However, when we looked at the subclasses, we saw that this IgG1, which is a Th2 polarization, was enhanced for the pDNA alone. In the experiment 1, we saw that the TMC DNA induced the reduction of IgG1 titers, so the opposite of what we saw in the second experiment. In the second one, TMC nanoparticles that led to an enhancement of especially IgG2C, so more like a Th1 polarization in this case. So in the first experiment and this experiment, we saw that the TMC DNA plus MDP increased the IgG to B and C titer. So it was towards a Th1 polarization again. We looked at the ratios between IgG2 and IgG1, and here you see again we get a polarization towards Th1 when we had the TMC DNA plus MDP in comparison to the pDNA alone, which gave us a Th2 response. Now, the hypothesis behind this, to explain this, looked into literature, and we saw, okay, high total IgG and IgG1 titers may be caused by the presence of non-transfectable DNA that is just present there at the injection site or the site where we applied this. So it was not associated to the nanoparticles. And this condition appears to be similar to that what you find in necrosis, where DNA is released outside the nucleus and outside the cell. So this gives the body or the immune system a sort of like Th2 signal, which was recently published also here in Nature Medicine 2011. Extracellular host DNA axis damage associated molecular pattern is linked to a Th2 bias of the subsequent adaptive response. So obviously that's something we see, so we need to take care what we do with the formulations of our DNA delivery systems, and this is why I gave the title Less is More. If we reduced the dose of the DNA vaccine on our nanoparticles, we could actually move the immune response into the right direction, namely Th1. So the conclusions from this, pDNA alone was shown to induce Th2 responses, while the particles polarize immune responses to Th1. 50 microgram of pDNA per dose absorbed to the nanoparticles is effective formulation to induce Th1 polarized immune responses, so more is less. Furthermore, the MDP, and we use this at 10 microgram here dose, further enhanced levels of IgG1, IgG2B, IgG2C, as well as the interferon gamma response in T cells. We did in the third study, we increased the concentration here of MDP, and we get again a polarization towards Th2 actually. So you have to find the optimal dose and the optimal ratio between the two PRR ligands when you are formulating your vaccine delivery system. 10 microgram MDP per dose incorporated into these TMC nanoparticles enhanced Th1 responses to TMC DNA. I'd like to thank a couple of people that were involved on the vaccine formulation laboratory connected to the WHO, present here at the University of Lausanne, BEI resources, 2 people from my group who helped in the lab, the Bioimaging Center, and we recently are involved in a cost action SimInhale, which is the bringing together drug delivery, pulmonary drug delivery to simulation of inhalation in terms of fluid mechanics. And I don't want to let you go without pointing out that we will have a workshop on the challenges and assessment similarity of equivalence of NBCDs in Baltimore, September 28, 29. It was jointly organized by UIPS, FDA, the Nanotechnology Characterization Lab, and the Non-Biological Complex Drugs Working Group. And I thank you for your attention.
Thanks, Gerrit, for sharing your interesting data and also keeping very much within the time frame scheduled. Maybe I can start as a chairman. The first questions. You see some trends with MDP or and also gamma interferon. But if you would like to design such a vaccine against tuberculosis, do you have a sort of reference criteria which you would like to reach? What are really the aims to strive for?
Well, that's a really good question. So what we look for is in the response in this animal model, and of course the decisive would then be the challenge test that we would do to protection against infection, which we haven't done yet.
Yes. Elias?
Yeah? Okay, thank you very much.
Oh, I didn't see that. I'm a bad chairman.
I can't see it because there's
Very
this
small.
light here in my face.
The light is...
So, I may have missed this, but I'm interested in how you think the DNA, where is it being expressed? Because our experience seems to be that DNA is destroyed in antigen-presenting cells, and that it's almost better to have it expressed somewhere else and then the protein be taken up into antigen-presenting cells. How does it work in your system?
Yes, we did several studies, in vitro studies. In vitro studies where we looked at this. This was an intramuscular injection, so possibly the vaccine was taken up by muscle cells and so forth. But we also had mucosal application of our nanoparticles, and we saw transfection uptake not only by macrophages or dendritic cells and a certain survival of the plasmid in these antigen-presenting cells, but also in epithelial cells where the protein then was also expressed.
So which one do you think is more important, or you don't know?
I would target the antigen-presenting cells.
So, following on this question, I don't know if you make the experiment. What will happen if you add your nanoparticles together with the plasmid DNA? Will you be able to revert the Th1 response?
So what you mean is that we only make a mixture of both?
Yes.
That's a good question. First of all, you would have to be quick not to have adsorption of the DNA to the nanoparticle surface. And then probably what would happen is that if this adsorption process would not be complete, what I would think is that you would get the same results as we've shown for the naked plasmid DNA alone in the absence of the nanoparticles. I would almost bet on this.
And another question, the particles change the shape and the morphology. Is this something that is a worry if you would like to register such a product?
In terms of manufacturing, I don't know. You have to see, you know, I can't say. I can't say. I don't know whether you want to have all the same shape and size of these particles. Size certainly, but shape, I don't know. But I think in manufacturing, you will not be able to completely avoid it. Elias.
Yeah, a quick one. Does it make sense to look at secretory IgA? I mean, in protecting the mucosal sites against infection?
We did this as well in the next experiment. That's something we didn't present today.
Okay. Oh, you did it?
Yeah.
And?
The secretion of IgA was not very high.
Maybe if you do it locally.
It was a nasal.
It was a nasal okay.
wash.
Okay. Thank you.
Last question.
My question is, BCG is not a very good vaccine against TB. In children, it protects to a certain age of severe forms of the disease, but not at all in adults. And what you picked is antigen 85A. It's an immune-dominant antigen in BCG. And you're suggesting that you can make a subunit or kind of subunit, but it's a DNA vaccine. I understand that. Wouldn't you make it actually worse by just picking one antigen from you see what I mean, right?
Yeah, that's a very good question. I don't think we make it worse. We also worked once with polyepitope DNA vaccine, which expressed a protein with different epitopes bundled together in one protein, which was supposed to make it more antigenic. We didn't see that, so we went back to the basics and we said, okay, we're using this 85A now as a single antigen in order to show the efficacy of our carrier systems, just to make the point there. But I agree, and I recently learned that there's a recombinant version of BCG now in testing in cancer, in bladder cancer. Actually.
Right, that's our study, by the way.
That's you, right. I can't see you here. It's
I'm going to present on that tomorrow afternoon
Very
if
good.
anybody's interested. So.
I'll be there.
So everybody heard that. Huh? So thank you, Gerrit. We move on